They are mostly the same questions that are in the easy generators. And my strategy is those self-grade, so you get the immediate feedback on that. The easy generators, you have to wait for me to approve the certificate. So that's why I'm having I turn that in last, and I'll just simply check your certificate to see if it's there. But you've already got your grade from the quizzes. So do the easy generators and then go do the quizzes and use that for practice. I'm going to ask that you do each of those quizzes at least once prior to the due date. But after the due date, you can do them again. There's no late penalty on them, other than the final deadline is going to be the morning of your exam. I mean, doing those homeworks after the exam, of course, makes no sense. It doesn't benefit you as much as doing them before the exam. So do them multiple times, but at least once prior to the due date, and then multiple times prior to the official final due date, which is the morning of the exam itself. All right. If you have any questions, of course, email me and I'll take it from there. Hopefully, this is pretty straightforward for you. And let me share my screen for you. All right. Get the chat box up so I can see any questions that come in. I do have a grade for the easy generator. It's just 10 points for the whole thing. So it's not broken out into the sections. So 10 points for the whole thing. I'm just going to look for your certificate. So please upload, because I was not planning on doing any grade transfers from easy generator, just to go through and just see if your certificate's there. But there's many, many more points coming from the questions, which are duplicated into the Canvas quizzes. All right. So there's 7 PowerPoints. However, the last 3 are all combined in as one large PowerPoint. And then the quizzes complement those 7 PowerPoints. I mean, those 5 PowerPoints they gave you, even though that 5th one is a triple PowerPoint. All right. So what I want to do is go through these slides. I've got 4 hours worth of slides here. I want to do this in much less than 2 hours, so I'm not going to go through every single slide, but I want to point out ones that I want to make certain that you're familiar with. If you need to reinforce this, I strongly encourage you to go to the textbook sections. Obviously, there's a lot more information in these textbook chapters than I'm going to cover, but they would be very powerful reinforcement for the stuff that I am covering. And a lot of my material comes from these chapters. So let's back up way back into just proteins itself. What are the 4 levels of protein organization? And hopefully remember this from BMS way back when. The amino acid sequence is the primary structure. Alpha helix and beta sheet would be secondary structure. The folding of the protein is your tertiary structure, and your quaternary structure is the assembly. Now, for hemoglobin, we're going to be talking about amino acid substitutions. So that means we have a misspelling, so to speak. Okay. All right, so we're going to talk about all the errors that take place in the primary structure, which are going to negatively affect the secondary structure, which will then affect the folding, which affects the overall tetramer. There's 4 subunits for hemoglobin, so the quaternary structure of hemoglobin is a tetramer. So the quaternary structure, the 4th level of organization, is 4 separate subunits coming together. A quaternary structure could be pentamer, trimer, dimer. So it's just assembly of multiple subunits. But for hemoglobin, it's 4 subunits, and they have to come together just right to carry oxygen appropriately. So I'm not going to be testing on this slide specifically, but just gives you an idea of what the spelling is of the myoglobin versus hemoglobin alpha, hemoglobin beta. Some more basics. Make certain you know what a peptide bond is. It is simply an amide bond where we take a carboxylic acid and pull off the oxygen, take an amine group, pull off the hydrogen or hydrogens, and lop off 2 hydrogens, 1 oxygen total for water to come off and the amide bond to form. Now, the peptide bond is an amide bond, but anytime we have an amide bond between 2 amino acids, for some reason, we also call it a peptide bond. I already mentioned that the secondary structure is limited to just 2 possibilities, alpha helix and beta sheet. And that's due to the fact that the peptide bond does not have a rotation on it. There's partial double bond character on the peptide bond, which limits the different possible structures that are available. You look at the figure of all the different possible angles that could be formed. All we have is what's in blue there, a limited number, and those are specific types of alpha helix and beta sheets. So because we don't rotate around the double bond or the peptide bond, which is actually a partial double bond, we have a limited number of possible shapes of the protein. All right. Folding, of course, is the tertiary structure. And we're going to chop up the tertiary structure into domains. So, for example, hemoglobin, sickle cell hemoglobin, has a defect at the beta 6 amino acid. Well, beta 6 falls into the alpha domain, so sometimes we call it the A6 amino acid. We're going to talk about other amino acids like, the G6 amino acid, where I forget what number total that would be, but there's two different addresses for amino acids. There is the overall address, where it is in the entire structure of 146 amino acids, or we can say which amino acid is it within any one domain. The folding is held together by a combination of salt bridges, hydrophobic interactions, meaning they don't want to be around water, so they fold in on themselves to get away from water. Hydrogen bonding is also a big factor. And I'm not going to circle this because in hemoglobin, there are no disulfide bridges, even though other proteins do have that. All right. And the quaternary structure is the fourth level of assembly. And by coincidence, the fourth level of assembly has 4 subunits. That's just purely coincidence. Okay. But the quaternary structure is the fourth level of organization. And it could be, a dimer, tetramer, trimer, pentamer. In hemoglobin, it's a tetramer. Right. So I flew through that pretty quickly because that was just a review of protein organization that was done in BMS a couple years back. All right, let's now move on to specifically hemoglobin itself. What's hemoglobin do? It carries oxygen. Primary function. Sorry about that. Still having problems with my electronic pen. All right, so the primary function is to carry oxygen. A secondary function is it doesn't carry all the carbon dioxide back to the lungs, but it can carry some of the carbon dioxide back to the lungs. And as an interesting side note, it also has a little bit of enzymatic activity where it generates some nitric oxide. And that nitric oxide causes vasodilation, which would make sense because the red blood cells want their blood vessels to be wide open so they can get through. In order to carry that oxygen, we need an iron, specifically iron in the +2 state. Iron's not water soluble, so we need a heme group to carry it. And hemes are not water soluble, so we need that globin protein, all those amino acids to carry the heme. And then we tuck that heme into the middle of the protein. All right, that's kind of a duplicate slide. And again, I'm going to cut to the chase here because I've been talking for 3 hours already today. This is now my 4th hour of lecture today. My voice is getting rather sore here. All right, so let me slow down on this slide and spend a little bit more time on this slide. The 4 nitrogens that are in the heme that are attached to the iron. So the iron is attached to 4 nitrogens inside the heme. This is not showing the entire heme, just the nitrogens inside the heme. It also attaches itself to a nitrogen in what's called a proximal histidine. There's another histidine called the distal histidine, which is not close enough to the heme to form a bond with it, which is perfect because what it does is it hydrogen bonds to an oxygen molecule that does coordinate with the iron in the 6th position. A better picture of it. And in domain F, 8th amino acid is a histidine that forms that proximal histidine bond. In the E domain, seventh amino acid. We have another histidine that forms a hydrogen bond to the oxygen, which coordinates with the iron. So just to visualize how these things all fit in and fit together. And then the oxygen is going to bind to the Fe+2, as we see in that picture there. I want to note that if the iron becomes oxidized to Fe+3, it can no longer bind to the oxygen. It's only the iron Fe+2. So we have enzymes inside the red blood cells that keep the iron in the +2 state. This is simply two figures fit together to kind of visualize how does that heme fit into the overall globin protein. This is a minor point, but just an interesting point, that we have two states of hemoglobin. We have the T state, meaning no oxygen. The R state is with oxygen. And when there's no oxygen, that means the iron is only bound to 5 other atoms. And it doesn't lie flat. It's kind of in a puckered shape. But when we have the oxygen bound to it, that means the iron has 6 bonds on it now. And it's in a flat planar state. So if you see the term puckered state, that generally means there's no oxygen. And we call it the T state. And that's called the T state because that's called the taut state. The R state is called the relaxed state, and that's because the molecule wiggles. Now, I used to do X-ray crystallography way back in graduate school, and if we were to crystallize a T state hemoglobin and put it in the X-ray machine and measure the location of the different atoms, you get some nice, precise locations of the atoms. But if you do oxyhemoglobin, you get a blurry picture because there's a little bit of movement in the molecule. And that's why it's called the relaxed state. That's where those names come from. But for you, just know that T state means no oxygen. R state means with oxygen. Right. There's some movement in the molecule. And I want to point out that the T and the R states, that the T is oxy. The T is deoxy. Let me get that straight. The T is deoxy. The R is oxy, and there's no in-between states. So let me get the rest of that figure up there. There's the T state, and there's the R state, and the shape of the molecule has a 15-degree shift between those shapes. So squeezing that oxygen in there causes a shift in the shape. And we call this cooperativity. Now, the reason why there's cooperativity is all 4 subunits shift at the same time. So we either have all 4 without oxygen or all 4 with oxygen. This figure over here says the same thing. It's all 4 without oxygen or all 4 with oxygen. And it doesn't like any of the possible in-between states. So that's called cooperativity. So it's kind of like the all or nothing. The reason for that is in the T state, there are certain amino acids that line up perfectly. In the R state, it's a different set of amino acids lining up. In between, nothing lines up, and that means it's a high energy state. So it either wants to be in the T state or the R state, and nothing in between. So either it has oxygen or it doesn't have oxygen. It's an all or nothing. Okay. So let's come back to where we're going with this. We're going to talk about the hemoglobinopathies. There's sickle cell anemia. There's a bunch of other rare hemoglobinopathies named largely from where they were discovered. And if there's this is just simply showing what's disrupted when we have what gets disrupted when we have one of these amino acid sequence changes. All right, so the T versus the R state. In the R state, it likes to bind oxygen. And we have 100% binding. When we go away from the lungs, go into the capillaries in your big toe or somewhere in any of your tissue away from your lungs, the oxygen pressure, of course, is going to drop. And the hemoglobin is pushed into the R state because of the oxygen. If some of those oxygens come off and we don't have the pressure to push them back on, the rest of the molecule is going to want to flip from the R state to the T state and drop to a lower affinity and let go of the oxygen. So about half of the hemoglobin molecules will get rid of all their oxygen. It then drifts out of the red blood cell, across the capillary walls into the cells that we're going through, and in the tissue, there's myoglobin, which always has a high affinity for oxygen. So if the oxygen goes into the cells are going to grab it with the myoglobin. Hemoglobin is going to let go. Myoglobin is going to grab it. So it moves from the capillaries, from the red blood cells in the capillaries to the myoglobin inside the cells, like in the muscle or the heart or the liver. Then that myoglobin will eventually give that oxygen to the mitochondria for the electron transport chain. So the physiology that's driving that is partial pressure of oxygen. So if oxygen in the lungs, we have high partial pressure, we're going to push the oxygen on, push the hemoglobin into the R state, and away we go. We get to distal parts of the body where the partial pressure of oxygen has dropped. If oxygen comes off, there's no pressure to push it back on again. It just then drifts away. And if we have a hemoglobin that's lost one oxygen due to cooperativity, all four subunits give up their oxygen around the same time. So the hemoglobins are either all or nothing, and about half of them give up all their oxygen. The oxygen diffuses because it's a small molecule. If it goes inside the cell, it's grabbed by myoglobin, and it does not go back to the blood. And then, of course, it's used by the tissue. BPG is a very important molecule. Is a -5 overall charge. Very, very negative. It binds in what's called the BP binding pocket of hemoglobin. And it stabilizes the T state. So BPG is blamed for causing hemoglobin for having a lower affinity for oxygen compared to myoglobin. Because BPG does not bind to myoglobin, it only binds to hemoglobin. So it induces hemoglobin to let go of the oxygen and give it to the tissues. Now there's a pH effect here because some of these positive charges are histidine. Histidine has a pKa of 6. That means if the pH of the blood were to drop a little bit, it becomes positively charged. If the pH goes up, histidine neutralizes. So what that means is as the blood becomes a bit more acidic, BPG binds more tightly, inducing hemoglobin to let go of its oxygen. Now, why would the blood be getting acidic in the capillaries? Carbon dioxide, when it dissolves in the plasma, turns into carbonic acid. That's going to induce BPG to bind more tightly to the hemoglobin. Hemoglobin is going to let go of its oxygen. Okay. Here are 3 disorders where the BPG site is being negatively affected by a mutation. Okay. The old name for it was DPG, but the new modern name is BPG. In experiments, and I've done these experiments myself because I used to do research on hemoglobin back in graduate school. If you had hemoglobin with no BPG, it would have the same looking binding curve as myoglobin. It's only when you add the BPG that it takes this S-shaped curve. And we have about 5 millimolar in a red blood cell. So hemoglobin only works inside the red blood cell. So the red blood cell breaks open, the hemoglobin doesn't function properly. All right. Which brings us to the Bohr effect. The Bohr effect is the higher the pH, the higher the binding. The lower the pH, the lower the binding. So why is that? Let's go back to this picture. The higher the pH, the less likely this histidine has a positive charge. The lower the pH, the more likely this histidine has a positive charge. And therefore, that's going to change how tightly that BPG is going to bind. And BPG promotes the T shape, which is no oxygen. So at lower pHs, BPG binds more tightly and the oxygens let go. So that's the perfect physiology. In the lungs, blood pH is a little bit higher. So in the lungs, pH goes up a little bit. Why? Because we're releasing carbon dioxide. Carbon dioxide, basically in water, is acidic. So if we're losing all this carbon dioxide in the lungs, pH shifts up a bit. The average blood pH is pH 7.4. But in the capillaries, we have less carbon dioxide coming into the capillaries from the tissue that lowers the pH. And what's happening is in the lungs, hemoglobin has a higher oxygen affinity. When we get to the capillaries in the presence of carbon dioxide, it has a lower oxygen affinity, which is exactly what we want. We want that hemoglobin let go of the oxygen and give it to the tissue. All right. As a side note, I see a student ask, can students do research with me? And the answer is absolutely yes. So I'll be more than happy to elaborate on that after we finish this section. All right, so continuing on, this is just simply another textbook saying the same thing, so I'm going to save you time. As I already told you, I've been talking for 4 hours already today, so I'm actually motivated to go quickly today. All right, so this is just simply restating what I said. As pH goes up, binding of hemoglobin goes up, which is exactly what we want in the lungs. And as pH goes down, which is what we see in the capillaries, binding drops. So this is inducing hemoglobin to bind oxygen tightly in the lungs, let go of it in the capillaries. So it's a perfect delivery device. So not only is carbon dioxide making the plasma more acidic, it can also bind to the hemoglobin itself. Form this carbamino group. And carbamino hemoglobin also does not like to bind oxygen. It pushes it into the T state. So carbon dioxide is pushing hemoglobin both due to pH and directly binding to the hemoglobin itself to let go of the oxygen. When we get to the lungs, because we're breathing in oxygen and able to breathe out carbon dioxide, that pushes these reactions back the other direction in the lungs. All right. Again, I'm going. This just shows that it's attaching to amino acid number 1. That's all. So it's the N-terminal amino acid that the carbon dioxide binds to. All right. Carbon monoxide. Why is carbon monoxide toxic? It binds iron more tightly than oxygen. Both oxygen and carbon monoxide like iron plus 2. Now, I don't have it on the slide, but what's methemoglobin? Methemoglobin is when the iron flips to +3 and no longer binds oxygen. So somebody who has a disorder called methemoglobinemia, they're anemic because their iron's in the +3 state and it's not binding to oxygen. Carbon monoxyhemoglobin, well, that's poisoning. When we have carbon monoxide substituting for oxygen on the iron. Any questions so far? Okay, very good. All right, so part four. I got 10 parts total here. All right, so I do want you to know the 6 different subunits that make up hemoglobin. Alpha 2 beta 2 is hemoglobin A1. Okay, so our normal adult hemoglobin has 2 alpha subunits, 2 beta subunits. Alpha has 141 amino acids, beta has 146 amino acids. So these are big proteins. Fetal hemoglobin, before you are born, you need a higher affinity hemoglobin because you're not using your lungs. You're getting your oxygen from the placenta. So you need a hemoglobin that has a higher affinity than your mother. So we substitute for beta the gamma subunit, which causes the fetal hemoglobin to have a higher affinity. Now, fetal hemoglobin is going to be alpha 2 gamma 2. Now, delta is high altitude hemoglobin. This is going to be alpha 2 delta 2. We all have a small amount of this, but it tends to upregulate if you're living at high elevations. If you're going to go climb Mount Everest, they make you stay at base camp for at least 2 weeks to get your hemoglobin to adjust. And these bottom two are only during the first 3 months of life, where we have I can't even draw the zeta. Okay, I'll just use a Z. Zeta 2, epsilon 2. That's your hemoglobin during your first 3 months of life. After 3 months, you switch to fetal hemoglobin. And after birth, you switch to adult hemoglobin. Right. So this just reiterates what I just said. Hemoglobin A1 is alpha 2 beta 2. Hemoglobin A2 is alpha 2 delta 2. That's for high elevation usually. Hemoglobin F is fetal hemoglobin, alpha 2 delta 2. And hemoglobin epsilon or hemoglobin embryonic is zeta 2 epsilon 2, only during the first 2 months, 3 months of life. What's interesting to you is a patient with sickle cell anemia will tend to have elevated fetal hemoglobin as a compensatory mechanism because the A1 is not working. A1 has been converted to sickle cell. Another interesting thing is we use hemoglobin A1 as a marker for diabetes. We take a blood sample and measure how much glucose is attached to your A1C. I mean, how much glucose is attached to your A1. C means conjugated. So how much glucose is conjugated to the A1, we'll call it A1C, and use it as a marker for diabetes. If you have more than 5 or 6% glucose attached to your hemoglobin, that's a concern because that means you may have that glucose attached to all your other proteins as well, especially in your kidneys, your retina, and other places that are vulnerable to diabetes. All right. So this is to reiterate that a fetus must have a higher oxygen affinity than its mother. So hemoglobin F has a higher affinity than hemoglobin A. And I have some practice questions in the online Canvas quizzes that will ask, rank these hemoglobins. And hemoglobin S would, of course, have the lowest affinity. Hemoglobin A1 would be certainly way higher than sickle cell hemoglobin. A2 is higher than A1. Fetal hemoglobin would be higher yet. And then above all of them would be myoglobin. Myoglobin, of course, is always way above all the hemoglobins because myoglobin has the desire to keep oxygen inside the tissue, pull it away from the blood inside the tissue, to inside the tissue. All right. All right, I'm going to come back to this, and I'm just going to gloss over this because I'm going to try to be as quick about this as I can. That during your first 3 months of life, you're getting most of your hemoglobin from the yolk sac, and that's the hemoglobin embryonic. Okay. And that sustains you until we switch to hemoglobin F. G is for gamma. This is kind of goofy the way they have it in that textbook. That's the gamma subunit, the hemoglobin with the gamma subunit. So it's hemoglobin F for fetal hemoglobin. And if you believe the picture as drawn, it looks like most of that's being made in places like the liver. But after birth, after 9 months, of course, we get our hemoglobin from our bone marrow, which does hemoglobin A1 with a small amount of hemoglobin A2. So A1 has the beta subunit, A2 has the delta subunit. The genes for these are on chromosome 11 and 16. And just as another side note, this information comes from a very famous author. Moving forward, this just simply restates what I just said. Which chromosome do the different subunits come from? All right, and this is kind of a repeat. Fetal hemoglobin, by necessity, must have a higher affinity than the mother. So, needless to say, hemoglobin F must have a higher affinity to take the oxygen away from the mother at the placenta because the fetus cannot use its lungs until it's born. As soon as it's born, it switches to hemoglobin A1. All right, that's the first PowerPoint. Any questions on that so far?